Hi everyone, this video is regarding DNA replication and how does DNA replicates. Back in 1953 when Watson and Crick published their DNA model, the question of how exactly DNA replicates still remained. There were three proposed models. The first one was the conservative model where you have a parent DNA produces two daughter DNAs. One has exactly two copies strands as the parent, and the other one had completely two new strands. The second one was the semi-conservative, where two DNA strands will separate, and each daughter one will have one strand from the parent, and the other one completely new one. The third model was dispersive. DNA will cut in various parts at the different places, and each will kind of look like a patchwork between the old and the new, and they're both, the daughter ones are the same. Meselson&Stahl's experiment was considered the beginning of genetic revolution. Many were skeptical that DNA separate because of how strong it was, and in order to determine which model was correct, they had to perform an experiment. They had to tag the old and the new DNA so they could compare them, and they did that with the help of E. coli bacteria. It was easier to grow and it replicated very fast, one generation every 20 minutes. Basically, they had to culture E. coli in heavy nitrogen medium for many generations until the heavy nitrogen was fully incorporated, and thereafter they had to transfer the heavy E. coli into the lighter nitrogen and observe each generation. They did that with the use of gradient centrifugation. Basically the density gradient, the heavy ones will be at the bottom and the lighter ones will be on top. With the first generation, they could easily eliminate the conservative approach because it yielded half of light and half of heavy. The next two divisions yield half of the light and half of light heavy or entirely light, so that eliminated the dispersive model, leaving the semi-conservative being the way DNA replicated. Their experiment not only confirmed the model, but it also confirmed that the DNA strands are separated by heat, and they're denatured that way. So very important experiment. Thank you.